Perfect. Hi, everybody. Thank you so much for having me. It's a real pleasure. I'm Simone, PhD student at the Children's Medical Research Institute in Sydney, Australia. And today we'll present some updates on our project towards a safe and effective gene therapy for SLC6A1, in line with the amazing presentations of this morning. I really don't have to explain anyone in the room that SLC6A1 codes for GAT1, which is a transporter that removes GABA from the synaptic space to allow its recycling. But I guess, again, in line with what we've seen today, we know that GAT1 is expressed mainly in a minor fraction of all our neurons and astrocytes in GABAergic inhibitory neurons. And therefore, in a gene therapy context, in an AAV-based gene addition context, we need to take in consideration the regulation of SLC6A1, how the gene expression is regulated. Just very basic slide. Gene addition strategy entails the use of an AAV, adeno-associated vector, that delivers a functional copy of our gene, SLC6A1, and if we administer this AAV into patients carrying a loss-of-function mutation SLC6A1, we aim to restore the physiological activity of GAT1 in affected tissues. Again, in our case, mainly the brain and mainly inhibitory neurons. And in our project, we aim to optimize the delivery of this transgene, of the therapeutic SLC6A1, and we aim to do that by working on specific elements of this therapy, the promoter of SLC6A1, the cassette, and the capsid of the AAV. Just very briefly, as we mentioned our speakers today, the SLC6A1 promoter regulates the spatial and temporal expression of the gene, where and when SLC6A1 is expressed, but also controls the strength of the expression in transduced cells. The cassette contains the coding sequence for SLC6A1, the instructions to make the gene and the protein, but also can contain additional introns, enhancers, and regulatory sequences that can influence the expression of our gene. And of course, the capsid is what packages and delivers the therapeutic transgene and controls which cells will be transduced. Starting from the endogenous promoter, we looked at public and available datasets like ChipAtlas to identify the best endogenous promoter candidate, looking at ChIP-seq, ATA-seq, and DNA-seq, all highlighting sequences in the DNA right upstream, the starting codon for SLC6A1, that have the correct signature to be called promoter. And we selected different versions of our endogenous promoter, identified a core promoter element that I called promoter 1, another a bit downstream, this promoter 1 that I called promoter 2, some enhancers present upstream promoter 1, and we are also testing a variant, a polymorphism, 21 base pair insertion that creates an enhancer element and potentiate the activity of potentially the activity of the SLC6A1 promoter, and it has been published a few years ago. We cloned all these different versions plus CBH control, which is a very strong promoter that expresses the gene ubiquitously. We clone all these different versions of the promoter, right upstream GFP. Now our promoters control green fluorescent protein expression. Again, to select in vitro, initially, the most specific and effective SLC6A1 promoter. I transduced with AAV7M8 some healthy donor, iPSC-derived human neurons to select the best performing promoter. And if we count the number of GAT1 positive cells and GFP positive cells, and of course the double positive GAT1 GFP positive cells, we can validate the promoter in vitro and measure efficiency and specificity of these promoters by measuring how many GAT1 positive cells are also GFP positive. We can measure efficiency. And of course, our controls, CBH, very strong and ubiquitous, and synapsin, pan-neuronal, are very efficient and target almost all our, 100% of all our GAT neurons, GAT1 positive neurons. And one of the endogenous promoters works quite well and targets almost all our promoters, all our cells. And if we measure the specificity, so how many GFP positive cells are also GAT1 positive, we can measure how specific is our promoter. And again, P1 and P2 are highly specific and show 85% of specificity. We are also planning, and we are testing in vivo our promoters in collaboration with the lab of Jean Paz in the Gladstone Institute. We are testing the promoters and some capsids, again, to select the most specific and effective SLC6A1 promoter that drives expression in target cells, as well as selecting the best capsid in our context through ICV injection in wild-type mice. We just got the samples literally yesterday, and I'm super excited to start analyzing them as soon as I come back. So yes, for the promoter 2 containing an enhancer and that 21 base pair polymorphism, we demonstrated that it is effective and specific in vitro, and the in vivo screening will allow to further validate the promoter and choose the best performing capsid among the selected. We are working also on the cassette, and we have made some variants of the cassette using enhancers and introns, different length of the 5' UTR, presence or absence of a small fragment of intron in the SLC6A1 coding sequence. We are testing non-codon optimized versus codon optimized sequences. We have cloned them under the CBH promoter and to select the most effective SLC6A1 cassette. And if we transfect all these different versions of the cassette in X930 cells, we see that the cassette number 6 is able to increase the mRNA levels, of course, of transfected cells quite nicely compared to all the other cassettes. The other variation of the cassette that we will soon test is the presence of a naturally occurring variant, a polymorphism, that is predicted to be non-pathogenic and allows for higher GAT1 activity. And the plan, again, is to screen between the best performing cassettes for the first screening and the second one. And as soon as we have selected the best performing promoter soon from the in vivo results and the best performing cassette, we will clone our, hopefully, final therapeutic transgene. And in collaboration with, again, the Paz lab in Gladstone Institute, we will verify the efficacy of our therapeutic cassette in the mouse models of SLC6A1. And we have just obtained also iPSCs, patient-derived iPSCs, and we want to test the efficacy of our therapy also in patient-derived organoids. And again, to finish everything, for the promoter, we have selected the best performing one in vitro, and we have to finish analyzing to select the best promoter in vivo, in mice, as well as the best capsid. For the cassette, we have selected the best cassette in vitro, and we want to test that non-pathogenic variant. And as soon as we have some characterization data from promoter and cassette, we will test in patient-derived models and mouse models. Okay, thank you so much to the foundations that allowed me to be here today, SLC6A1 Connect and Kids, as well as my lab, Leszek PI, Andrea and Ardik working on the project as well, and the Gladstone Institute, Pat, Julia, and Brett. Thank you so much, and thank you so much.